Alright, okay, so hello dears. Ayan, so hello once again and welcome to the third video and hopefully the last of our lecture on immunoassays for human parasites. And for this lecture, we're going now to discuss the last type of immunoassay, and that is your enzyme immunoassays. Right? Now for enzyme immunoassays, they follow the same principle with your fluorescent immunoassays. But the difference lang is, instead of using fluorescent fluorochromes or fluorophores, we use now enzymes. Ayan. So the antigens and antibodies are labeled with your enzymes. Okay, so sana all may label. Okay, yes, more in your IAS in third year, we will discuss these labeled immunoassays. Mga gusto naman label dyan. Okay, so your enzyme immunoassays and fluorescent immunoassays are examples of labeled immunoassays because they use labels. Ayan, so mga gusto ng labels dyan, baka labeled immunoassay kayo. But if not, baka mga precipitation agglutination lang kayo. Joke. Anyway, Mark, whatever. Okay, so again, instead of fluorochromes or fluorophores, similar to your fluorescent immunoassays, for enzyme immunoassays, you use enzymes, okay? And another name for that is your ELISA, alright? ELISA, enzyme-linked immunosorbent assays, okay? Alright, now the most common enzymes used are your horseradish peroxidase and ALP, alkaline phosphatase. Ako press the buzzer na, alright? Horseradish peroxidase, kalamunggay, alright? And alkaline phosphatase or ALP, alright? So these are some of the enzymes, the most common enzymes rather, that are used for enzyme immunoassays. Okay, alright. So after binding of an antigen and antibody takes place, you add a substrate. Diba? Because again, recall in your organ, biochem, an enzyme binds to a substrate. Okay, that's the partner of an enzyme, it's the substrate. Okay, and when the binding of substrate, when there is an addition of substrate, you then look for a color change. Okay, and the intensity of the color change will correspond to the concentration of the antigen and will often indicate a positive result. Okay, and the intensity again of the color proportional to the concentration of the antigen-antibody complex. Okay, so here's an example of an enzyme immunoassay. So example your antigen is in solid phase again, could be in tubes, microtiter plates, microtiter wells, plates, wells. So kabante mo itong mga yung circles here. Dito siya katundulaan. Ayan. So these are example of microtiter wells. Okay, microtiter wells because they utilize small volumes. Okay. All right. Ayan. So example in this case, solid phase antigen meaning the antigens are attached on the surface of the wells. Okay, alright, and you're detecting for patient antibody. Okay, so of course, once you add the patient antibody or the serum to the wells or to the tube, of course, they will bind. Alright, and of course, they're still washing to remove the unbound antibodies. So there's already an antigen-antibody complex. You then add your, again, enzyme-labeled anti-immunoglobulin. So again, it's an antibody against an antibody, but it has a label, an enzyme label attached to it. Okay, alright, so these enzyme labels, alright, will then attach to the antibodies and in the immune complexes. And of course, once you add the substrate, okay, you then examine for color. Okay, and again, the intensity of the color is proportional to the amount of antigen-antibody complex or the antibody concentration. Then, alright, okay, so this is one of the formats of enzyme immunoassays. There's a lot of formats for immunoassays. For enzyme immunoassays, I just chose this one. Okay, alright. So again, it depends on what you are identifying. Okay, in this case, we are looking for patient antibodies, but you can also look for antigens of the parasite, etc. Whatever, depending again on your need, depending on what you want to detect. Okay, alright. So those are the main immunoassays that are commonly used for parasites. Okay. Now we go now to mga other topics lang na ginagmay that usually use mga serology or your antigen-antibody reaction. And the first one are your skin tests for human parasites. Now your skin tests are performed by injecting parasitic antigens intradermally in the skin of your patient. Okay, so I don't know if you have experienced this, but skin tests are similar to if kanang you are to, if performs a doctor here, if he or she wants to detect if you're allergic to a drug, diba? So he or she performs a skin test, diba? Usually here, I tried that. If you are allergic to a drug, okay, so a portion of the drug is injected to your skin, and then if there is a wheal and flare, or like there is a naimagutod or naimagreddish, then it indicates that you're allergic to the drug, parang ganun, okay? And that is the same principle here, alright? Usually your skin tests are performed for allergies, okay? But we can also use that for other pathogens, parasites, and especially very common then, aside from parasites, is for tuberculosis, your Mycobacterium tuberculosis skin tests, okay? Alright, now again for parasites, we inject a parasite antigen intradermally and observing the reaction. If there is an immediate hypersensitivity, or it also means allergic reaction, a wheal and flare response is seen within 30 minutes na, so very fast. But if there's this is an example of a wheal and flare, so wheal, ayan, wheel, 5,000, joke. So wheal, meaning there is parang a, nainibutod, okay? There is parang an elevation, parang ganun. And then the flare is the color red. So that's the wheal and flare reaction. And this indicates a positive result for a skin test, okay? Or allergic reaction. So this is a positive result for a skin test. Now there are some skin tests that look for delayed hypersensitivity, meaning it's a type of allergic reaction or a type of hypersensitivity reaction that occurs after mga pila pa ka-hours, okay? And we look for erythema and induration. Erythema meaning reddish, nag-red ang area, and induration, much bigger na elevation, mas bigger siya na pagbutol, okay? And sometimes they can also be blister-like. It can result to blisters, okay? So here's an example of induration, and it's much bigger, okay? And for a positive result, we usually measure the diameter of the induration, okay? Alright, so that's for example of skin tests, okay? Alright, again, we perform it for parasites. Very common use is for allergic reactions, and again, also very common for your tuberculosis infection, okay? To detect if you have been infected with tuberculosis, or are you having a current tuberculosis infection. Okay, alright, ayan. So here are some of the skin tests in your parasitology for the different diseases. So very, kailangan lang yun ninyo ma-learn, or what you need to emphasize lang is the names, okay, for the different disease. So for ascariasis, you have the Mohn test. Sana all. Okay, alright. Next, you have the hydatid disease, which is your Casoni's intradermal test. Trichinosis or trichinellosis caused by Trichinella spiralis. Your Bachmann intradermal test. Kala-Azar Dubin fever, you recall in our Leishmania Dunovani lecture. The Montenegro or Leishmanian skin test. Toxoplasmosis, Frankel's or Toxoplasmin test. And for schistosoma or schistosomiasis, you have Fairley's test. Ayan, so you need to memorize, okay? Alright, so these are just some of the name of the skin tests, but for the other diseases, non-parasitic, there are still other skin tests for the other non-parasitic diseases. So what I just put here are the parasitic diseases, okay? Alright, so please be familiar, alright, your skin tests, okay? Alright, now next for the skin tests are what we call your rapid antigen detection systems, or your rapid tests. Your rapid tests follow the principle of immunochromatography. So those are your test kits. Okay, so recall in your test kits, diba? I will not discuss muna the principle of immunochromatography, but immunochromatography is the principle of your test kits. Immunochromatography. Okay, so this is again another type of immunoassay. Alright, this is an immunoassay that is found lang in your test kits. Your test kits, mga good, are very rapid, they're very available, convenient, and they offer accessibility, okay, to testing in the homes. Actually, your test kits are designed primarily for testing at home, okay, for your POCT or point of care testing, diba? Point of care testing, or your POCT. Most of your test kits. And your test kits are designed primarily, which is the main reason for POCT, point of care testing, meaning outside the lab, in homes, or other other people aside from lab personnel can perform the tests. Okay, all right, and that's POCT. Now, these are your rapid antigen, pwedeng antibody, rapid detection test systems, RDTs. Again, immunochromatography. Okay, that's a type of immunoassay. Your test kits. Okay. Alright. Now your RDTs or rapid tests, they detect soluble proteins, antigens, di ba? Through their ability to bind, to capture antibodies contained in a nitrocellulose strip. So inside your test kit, dears, you have a nitrocellulose strip there. Okay, and in the strip, in the nitrocellulose, inside the test kit, it has already antibodies that is on the surface. Okay, so they're now ready to capture antigens found in your sample, in your serum. Okay, alright, ayan. So the clinical sample is placed on the strip, and then we add a buffer, and then of course we then look for a colored band. Okay, the positive result for your test kits are usually your colored band. Diba? And always take note when interpreting test kits. Always make sure that there is a line on the control line, okay? Because if there is no line in the control line, even if there is a line on the test band, diba? Test kit, you have the frame here and then the control and test line. If there is no line on the control pero na ay line sa test, that is still considered invalid, okay? Always take note that whatever the result is, there should always be a line on the control band or in the control region. Why? Because that signifies or that indicates that your test kit is working properly. Okay? Because all the reagents are able to react in the control. Okay? So whatever the result in the test band is, you are confident enough that the result is really accurate, that the result is really correct. Okay? Alright. So again, interpreting test kits if there's no line in the control line, always invalid. Okay? Alright. Ayan. Okay. Now, here are some examples for Georgia and Cryptosporidium. These are brands. We have a test kit that it's a test kit that can detect already both Georgia and Cryptosporidium. So it detects Georgia and Cryptosporidium antigens, okay, in one test kit, diba? So 2 in 1, hitting 2 birds with 1 stone. Alright, okay. So expert Georgia Cryptosporidium assay. For Enzyme we have different ways. First, you have the RIDA screen, which detects IgG antibodies. And you have the Prospect Entamoeba histolytica microplate assay. So it's an example with your, it's similar with your microtiter wells, okay? But this time, it detects your Entamoeba histolytica specific antigens or EHSA in human fecal samples. Alright. And for malaria, very important, tumalabas sa boards, your optimal assay. Your optimal assay detects parasitic LDH or lactate dehydrogenase, which can detect, or it's used to detect all Plasmodium species. Alright? Optimal, okay? Optimal. It detects parasitic LDH. It follows the principle that malaria, your Plasmodium species release, okay, LDH, release LDH in their life cycle, no, in their destruction of the RBCs. Alright? So once parasitic LDH is detected in the system, then it indicates that the patient is having a Plasmodium infection. Alright? And the LDH of the parasite is detected if the amount of parasites in the blood is about 100 to 200 parasites or Plasmodium per UL of blood. Please take note. So the parasitic LDH sensitivity of the test, optimal. Alright? Parasitic LDH can only be detected in the body or in the blood if the parasites or the Plasmodium is about 100 to 200 per IU per UL of blood. Alright? Okay. So if the amount of parasites are not in this range it's below below, then of course the LDH cannot be detected. So it should be 100 to 200 or above. Okay? But the range, the minimum range that the test can detect LDH is if the blood already contains 100 to 200 parasites per UL of blood. That's the minimum. Okay? Alright. And it's optimal assay. That's the first. Next, we also have the Malaquick and Parasite F. Now, Malaquick and Parasite F, it's now very specific now for your Plasmodium falciparum because it detects an antigen of Plasmodium falciparum, which is your HRP2 or histidine-rich protein 2, which is only found in Plasmodium falciparum. Okay? So Malaquick, Parasite F, very specific, only for Plasmodium falciparum. Okay? Why? Because it detects your histidine-rich protein 2 antigen, HRP2 antigen. Okay, and the last one is the Binax Now Malaria test. Again, these are brands. Alright. For Binax now, it detects HRP2, which is specific for Plasmodium falciparum, and at the same time, it also detects a pan-malarial antigen, meaning it's an antigen common to all of the 4 species of Plasmodium. So it can detect specific for Plasmodium falciparum, and it can also detect even mga other infections caused by the other Plasmodium species. Alright? Ayan. So that's for the Binax now malaria test. Alright? Please take note, lumalabas sa boards. Promise. Okay. Not in my board exam, but in previous board exams. Alright, okay. Ayan. So here's an example of a picture. This is your Georgia crypto. So as you can see, there's a control line. There's a line of control, a line of Georgia, and line of crypto. So in this result, it means it's positive for both Georgia and Cryptosporidium na infection ba. Alright? Okay. And here's an example of your microplate assay. As you can see, diba? These are wells. Okay? So these are parang mga suludlan. Okay? They are containers in a way. They are wells. So nasa depression. Alright? Ayan. So these are the colors. Again, these are in a way follows enzyme immunoassays pa rin. So we look for color changes. Okay? Alright. Ayan. So that's for Prospect E Histolytica microplate assay. And then we also have here your Malaquick, the lines, and then you have Binax now. So as you can see for Binax now, you can detect for Plasmodium falciparum and for other Plasmodium species. Okay? So for T1, it's Plasmodium falciparum or mixed. Okay? So you have the guide there on how to interpret sa mga lines. Alright? But ang point lang, Binax now, it can detect both Plasmodium falciparum specific or other species of Plasmodium. Alright? Okay. So for LDH, sino nga yung malaria test that detects LDH? Your optimal assay. I don't have a mnemonic for that. But I think your optimal ADH, optimal ADH. Okay. And then for Malaquick, so it's very specific. Quick to identify P falciparum. Parang ganun. Okay, you can formulate your own mnemonics there. Okay, Malaquick, optimal. Alright. Okay, now we go to this very specific test, the Sabin-Feldman-Dye test. This test is only performed, it's the only serological test, the first serological test for toxoplasmosis. Okay, so it's used for diagnosis of toxoplasmosis, and the gold standard, the reference method for the detection of IgG antibodies to your Toxoplasma gondii. Alright, the principle is complement-mediated neutralization. Alright, so how does it work? So first is we then incubate muna live tachyzoids. Diba, recall in Toxoplasma gondii, the tachyzoids are the parang motile form yata and the active form of Toxoplasma gondii. Diba? So these tachyzoids are first incubated with a complement. Diba? Recall purpose of complement, and your test serum or the serum from the patient. Alright. Now, we then, after incubation, we then add your alkaline methylene blue, alright, methylene blue, which is a dye, and then we incubate. Now, in the case that the patient has antibodies, okay, to Toxoplasma gondii, the antibodies found in the test serum will bind, okay, so example, these are tachyzoids. Sorry naman, okay. So let's say your serum has antibodies, alright, to Toxoplasma gondii tachyzoids. The patient's serum has antibodies to tachyzoids of Toxoplasma gondii. Alright, so they will bind, of course. Now, diba, there is an antigen-antibody complex. Now, recall that there's also a complement in the system or in the test system. So, therefore, the complements will then be activated and will bind, diba, will bind to the antigen-antibody complex. And what happens, diba, if the antigen-antibody complex or if the complement is activated, the cell will lyse. So therefore, so this is now your lysed tachyzoids, diba? Because again, the complement is activated. Alright. Now, because the tachyzoids are already lysed, therefore, the alkaline methylene blue cannot already be absorbed by the tachyzoids. Okay? Therefore, the positive result is, of course, tachyzoids did not absorb the dye, meaning they remain colorless. And negative results, negative for antibodies, the tachyzoids absorb the dye. Okay? Because again, neutralization, meaning the antibodies has already been, the complement, no? Or the tachyzoids already been lysed or has already been neutralized. Therefore, it cannot anymore absorb the dye. Okay? Because again of the attachment of the antibody and the activation of the complement. So because of the activation of the complement, the tachyzoids will lyse, okay? And therefore, buslot na sila. they already have holes. Okay, so therefore they cannot absorb anymore the methylene blue dye. All right, that's the Sabin-Feldman dye test. All right, so I hope you understood. Okay, so this is the result. Positive result for Sabin-Feldman dye test. Again, the tachyzoids remain colorless because again, they were not able to retain or they were not able to absorb the dye, which is your methylene blue, because again, the patient has antibodies against the tachyzoids, which led to the antigen-antibody complex formation and the activation of the complement. And because during activation of complement, it results to the lysis of your tachyzoids. Okay, and because the tachyzoids have already been lysed, they cannot anymore absorb the methylene blue dye. Alright? Okay, so that's for positive. It means the patient has antibodies against Toxoplasma gondii. And lastly, these are your negative results. These are your lysed tachyzoids. And these are your negative results. As you can see, the tachyzoids has absorbed, or they have absorbed the methylene blue dye. Alright? Okay, so meaning the patient doesn't have any antibodies to Toxoplasma gondii. So that could indicate that the patient doesn't have or did not have any infection against Toxoplasma gondii. Alright, so kaya wala man siya antibodies because the patient doesn't have antibodies. Alright, okay. Ayan. And of course, yeah, these are your tachyzoids that absorb them. Alright, okay. And of course, for high-end laboratories or for other laboratories, not so much in routine laboratories, we can also perform your PCR and molecular assays. Alright, usually through nucleic acid hybridization. We use nucleic acid probes, which are small pieces of your DNA or RNA that is complementary or similar to the nucleic acid of the parasite. And of course, we use PCR. PCR is the amplification of the DNA of the parasite, alright? So that it's much more easy to detect. It's much easier to detect. And we have 3 stages. Akong mnemonic is dye, like imuhang beshi. So it starts with the denaturation. You have annealing, and then lastly is extension. Dye. Like how you call your friends. If you call your friends that dye, denaturation, annealing, and extension. That's the stages of PCR. Ayan. So denaturation, annealing, and extension. Dye. Okay, DAE, dye. Alright. So that's for molecular diagnosis. Again, personally, my knowledge on molecular diagnosis is very low. It's very little, and it's not stable. So yeah, I'll not dwell more on this. Okay, but you'll have a subject in your 3rd year, molecular diagnostics, more methods, molecular methods will be introduced there, and in-depth discussions will also be introduced there. Alright, so in your 3rd year, 2nd sem. So, andam now. Alright, okay. Ayan. Alright, so that's for molecular diagnosis. And lastly, for a summary, this is from your Zyvig. So as you can see, the PCR, the molecular assays, usually are really the ones that can provide the more definite identification of the parasite. Because again, you're looking now at nucleic acid, genetic material of the parasite. Alright? But again, for serological tests can still be very much helpful. Alright, especially again, as we have mentioned in the first lecture, especially when the parasites cannot be demonstrated or they are not easily seen in clinical samples. Diba? So we rely on serology or for the testing of the patient's serum for either antibody or antigen of the parasite. Okay? Alright, and here are your different tests. Again, for CA here, it means card agglutination. It's an agglutination that is in a paper or plastic card. Alright, again, that's based on manufacturer. Your plastic or your paper card is where you put your reagent and then you examine the agglutination there. Okay? Alright, rapid, meaning rapid tests. Okay, latex agglutination, again, latex agglutination, it uses latex particles as the carrier particle for your antigens. Okay, alright, ayan. So basically that's the summary. Again, that's from, this is from your, from Zyvig. Okay, alright. And that's the end of everything, actually. So this is the end of our lectures in parasitology, laboratory, and then of our class in parasitology. Laboratory. It's very, again for me it's very sentimental whenever I end my series of lectures, pre-recorded lectures, because again, under normal circumstances I would have really looked forward to this year of my teaching. This is my first year of teaching now, going 2 years now. Oh my God. Alright. But yeah, especially last time I was really looking forward to because I was able to handle my most favorite subject, which is bacteria. I was really excited to teach my students back then how to culture, how to identify bacteria, but because of COVID and all that, so wala. And now this again for parasitology, which is another one of my favorite subjects, which again you were deprived of skills and all that. But again, whenever I end my lectures or my series of lectures at the end of the semester, my only goal talaga, which again I have already emphasized at the start, is that at the end of our class, no, at the end of it all, you will be able to look back in the future in our class and say to yourselves na, okay, I already know this. Yeah, I've already learned this. I remember this from my class with Sir Mark. This has already been discussed. It will be very easy now for me during review back. Because again, I already understood or I already know this. Okay, so it will only be a review. Okay na talaga. So and I will not have a hard time. Yung talaga, more dyan na siya akong goal actually in all of my classes, especially with parasitology back then because these are my favorite talaga. Yeah, I apologize if there have been mga concepts that I was not able to discuss properly, but I just hope pa rin that most of our concepts you still remember or you still learn. And please talaga, I highly emphasize, do not forget everything, especially with the names of the parasites, the common names, infective stage, and the different methods, laboratory methods, and these also immunoassays, although this is the last and maybe major complex on topics. But again, I tell you, you will encounter this again in the future, especially when you become third-year students, interns, and even in the review for board exams, alright? So, I thank you so much, dears. I can see all your efforts. Thank you so much. And you give yourself a pat in the back and a round of applause for really surviving the semester and even this school year of online classes. We can only hope talaga that next school year will be face-to-face na, especially that you will become third-year students na. Majors, most of the majors for medtechs, in medtech are in third year na, so you need all the skills, the laboratory exposure that you need. So hopefully na talaga. But again, in our class, I just hope na talaga na nalingaw ra mo, that nalipay ra po mo na you were happy that I was your teacher for this sem, because I'm really happy that you are also my students. Yeah, so I hope lang good that you appreciate everything. I appreciate all your efforts. And thank you also for your kind words, especially if you say that I see some reactions that, you know, my lectures are very effective in a way. So, nakakatawa ng puso actually. We need validation. I need that. Okay? I need validation. Pero thank you. You are really the reason why I continue to teach. Bahalag kapoy na. Bahalag po sa hiding ng sugarahan. Okay? Or, yeah, because again, I plan to really have this sem as my last sem as a teacher. But I don't know with time. parang malilihis tayo sa plano. It's all in God's hands. But anyway, alright, so that's the end of our class in parasitology and the end of all of our lectures. Thank you so much, dears, sa lahat. Especially for you guys, for dears who are really watching talaga and listening. Thank you so much. Again, I hope you learned something. Thank you so much. Again, I hope you learned something talaga and you retain something from me. Alright, so thank you so much, dears. No, have a great day, and I'll see you whenever I see you. Keep safe always, and get vaccinated if you have the vaccine. Alright? Thank you so much. Have a great day.